These are the second part of Unit 6 notes, which covers mitosis, which is division of the cell. We'll start off with a video. There are a couple of videos in the set of notes, and so we're going to start off with the video talking about how DNA coils to make the chromosomes.
The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. Chromosomes are not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
So as you're looking at the structure of DNA, here's a chromosome, the familiar chromosome that you're accustomed to seeing. Realize that this is made up of coils, made of other coils, made of the double helix of the basic DNA structure. Wrapped around the histones, coiled up. Those coils are coiled up even more and even more to make the chromosomes. Chromosomes are not visible in the cell until time for cell division. Most of the time you see chromosomes in the nucleus as just chromatin because the DNA is spread out because it's being read and copied, and sections of it are being copied to give instructions to make proteins. Only during cell division is it necessary to coil the DNA up tightly into the chromosomal bodies that you can see. Another thing I want to point out to you is that a chromosome, as you're accustomed to seeing this X-shaped chromosome, is really made of two separate sister chromatids. This is what DNA looks like. This is what the chromosomes look like after replication has taken place in the S phase of the cell cycle. These two halves, each one is circled in red, make up one chromosome, but those are going to separate. They're attached to the centromere here in the center. Sometimes it's at the end, depending on the chromosome. But when cell division occurs, these two are going to separate from each other. These are identical chromosomes that will each go to one of the daughter cells that are produced in the process of mitosis. Mitosis has four phases: prophase, metaphase, anaphase, and telophase. The phase part is easy to understand. Pro means beginning, meta means middle, ana means apart, and telo means end. And so this tells you kind of where these fall in the process: prophase, metaphase, anaphase, and telophase. So this video is going to be about the phases of mitosis. It's just a little intro about the phases of mitosis with some pictures, and then we'll talk more about the details of what happens in each phase.
The cell cycle consists of 5 main sections. G0, G1, S, and G2 are all part of interphase. The actual division of the cell occurs in the M phase, mitosis. Mitosis can be divided into 4 stages: prophase, metaphase, anaphase, and telophase. The first stage is called prophase. During prophase, the nuclear envelope, which contains all genetic material for the cell, begins to dissolve. The DNA within the envelope condenses to form chromosomes, as pictured here. Centrioles with radiating spindle fibers migrate to either pole of the cell. The second step is metaphase. During metaphase, homologous chromosomes line up along the middle of the cell. This imaginary wall is often called the metaphase plate. The spindle fibers attach themselves to the middle of each chromosome at the centromere. The centromere provides a sturdy attachment point among the fragile DNA. Here is another depiction of metaphase. Next is anaphase. During anaphase, the chromosomes are pulled apart from each other towards opposite poles via the spindle fibers. This process can be divided into two sections: early anaphase and late anaphase. The last step in mitosis is telophase. During telophase, the cell membrane or wall begins to cleave between the separated DNA. At the same time, nuclear envelopes begin to reform. The result of telophase are two genetically identical cells, almost completely separated and preparing to go back into interphase. After mitosis is complete, the final product is a cell that is the exact replica of its neighboring cell. It has a complete nuclear envelope formed around the unwound DNA. Let's review the steps, shall we? We have prophase, metaphase, anaphase, and telophase. Look at this chart and see if you can match the pictures with the phase names that we just said. Could you identify them correctly? Hope so.
So let's talk about each phase in a separate and talk about what's happening. In prophase, this is when you can identify prophase in looking at cells when you can see the chromosomes inside the nucleus. In animal cells, the centrioles, which organize the microtubule network that's going to separate the chromosomes, separate and start migrating to opposite sides of the nucleus. The nuclear envelope starts to disappear as it's being reabsorbed by the ER. Remember that there are no centrioles in plant cells, but the spindle does start to form in a similar way at that time. In metaphase, the chromosomes have lined up in the middle of the cell, in the center of the cell. Each one of the chromosomes is attached to two separate spindle fibers, one from each end of the spindle, one from each pole of the spindle. This is important. They're going to attach there at the centromere, and they're going to create basically a tug of war between the two poles of the spindle. In anaphase, the centromere split, and just like in a tug of war, if somebody cuts the rope, both sides are going to fall back, and that's kind of what happens here. Since the chromosomes are being pulled on by both ends of the spindle, then when the centromere split, the chromatids are going to separate and be pulled apart toward opposite poles. At the same time, you can notice that the cell starts to stretch because some of those spindle fibers are also pushing against the ends of the cell to stretch the membrane. And the final phase is telophase. In telophase, the chromosomes begin to disperse again into chromatin rather than being in the chromosomal bodies. The nuclear membranes reform around each nucleus, usually at the same time as or immediately following telophase, cytokinesis occurs. This is the splitting of the cytoplasm. And you're going to end up with two cells that have the same number of chromosomes as the original cell had. They're basically clones of each other. At the end of this whole process, you have two daughter cells that are identical to each other, and each one is going to start its own new cell cycle. Now, cytokinesis, division of the cytoplasm, happens a little bit differently between animal cells and plant cells. In animal cells, you have the development of a cleavage furrow. It's basically fibers of a protein called actin that kind of act like a drawstring to pinch the original cell into two daughter cells. It's a gradual process. It happens relatively quickly, but it's going to pull those two things apart. Basically, they pinch apart into two separate cells. In plant cells, it happens differently because plants, remember, have a cell wall. And so they have a formation of a cell plate, which forms between the two new daughter cells. It looks a little bit different. Rather than pinching off, you have the development of this cell plate in between that will separate the daughter cells from each other. There are videos following this that are going to show you animal cell cytokinesis and plant cell cytokinesis. Now, you also have to remember the phases of mitosis, and you have to remember them in order, not only what happens, but the order in which they happen. And so a way to remember this is to come up with a sentence to remind you. Here's one to help you remember the stages. Pat my alligator tenderly. P stands for prophase, metaphase, anaphase, and telophase. If you were in class, I showed you the hand signals to help you remember what happens. If you were not in class, remind me and I will show you how. It's very, very easy, or you can get another student to show you. All right, here's a video about animal cell mitosis and cytokinesis.
Differential interference contrast microscopy is used here to visualize mitotic events in a lung cell grown in tissue culture. Individual chromosomes become visible as the replicated chromatin starts to condense. The two chromatids in each chromosome remain paired as the chromosomes become aligned on the metaphase plate. The chromatids then separate and get pulled by the mitotic spindle into the two nascent daughter cells. The chromatin decondenses as the two new nuclei form, and cytokinesis continues to constrict the remaining cytoplasmic bridge until the two daughter cells become separated.
So you can see that the cytoplasmic bridge occurs there and that the constriction occurs, kind of pinching off the two cells separate from each other. Next we have a video about plant cell division, showing the formation of the cell plate.
As this plant nucleus, taken from a blood lily, prepares to divide, the chromosomes first condense during prophase. Next, they gradually line up across the center of the mitotic spindle. At the metaphase to anaphase transition, the sister chromatids of every chromosome pair separate suddenly, in striking synchrony. The chromosomes are pulled along the microtubules of the spindle to opposite ends. After chromosome separation, small vesicles line up in the center of the spindle and fuse with each other to form a new cell wall between the two daughter nuclei. At telophase, the chromosomes decondense in the newly formed nuclei.
So you can see the difference between animal cell cytokinesis and plant cell cytokinesis. Next, we have a video that shows the phases of mitosis and what's happening to the chromosomes, just as a review of the whole process of mitosis.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
This concludes notes on mitosis.